Hi guys, welcome to Chapter 15, Part 2, Section 3 through 5. We're going to start with talking about Chapter 15, Section 3. Linked genes tend to be inherited together because they are located near each other on the same chromosome. Alright, so genes located on the same chromosome that tend to be inherited together are called linked genes. There's a difference in sex-linked genes and linked genes. Sex-linked genes is one gene linked to a sex chromosome. These are two different genes, two different characteristics that are linked to one chromosome, and they're thought to be inherited together because of where they occur. Alright? Morgan, our Drosophila experimenter, did some other fruit fly experiments to see how linkage affects the inheritance of two characters. He crossed flies that differed in traits of body color and wing size. Okay, so this is showing you the parents in his test cross. He took true breeding for gray body, normal wing style, and true breeding, that's the female, and true breeding for black bodied vestigial wing style, mutant type. And he crossed those, and the parent here is what you got from that first cross. So this would be your F1, and this was the test cross because he wanted to see what the offspring of this cross would be. So this is a heterozygote that is gray with normal wings. Okay, the wild type, gray with normal wings, crossed with a mutant black fly with vestigial wings, and most of your offspring end up either heterozygous for gray body, normal wings, or homozygous for black body, vestigial wings. Okay? Alright, what he found in his experiments, so here's your two pure breeding. They received this F1, he bred it to another double mutant, and this is the outcome of the offspring. Alright, here's your predicted ratios. They predicted if they're on different chromosomes that you would have a 1:1 to 1:1 ratio. If they are linked chromosomes on the same chromosome and they're always inherited together, if it's a linked gene, you would have all wild type and or all black vestigial. There would be no mixing of the two. However, what they found was a higher occurrence of the standard wild type and the standard mutant. However, they did experience wild type body color with mutant wings, mutant body color with wild type wings. Okay, so this was not eliminated as they predicted. So, while those two genes are thought to be linked, there's something else going on as to why those genes don't always travel together. Okay? So that was his point of focus and what we'll talk about in the next two sections. Alright? Even with linked genes, non-parental phenotypes can be produced. They were produced. Understanding this result involves exploring genetic recombination, the production of offspring with combinations of traits differing from either parent. Genetic findings of Mendel and Morgan relate to this chromosomal basis of recombination. Alright? Recombination of unlinked genes, you have independent assortment of chromosomes. Alright, Mendel observed that combinations of traits in some offspring differ from either parent. Offspring with a phenotype matching one of the parental phenotypes are called parental types. Offspring with non-parental phenotypes, new combinations of traits are called recombinant types or recombinants. A 50% frequency of recombination is observed for any two genes on different chromosomes. Okay. Here is an example of parental types versus recombinants. This figure is showing gametes from green wrinkled homozygous recessive parent and a gametes from a yellow round heterozygous parent. yellow round being your dominant traits, green wrinkled being your recessives. Okay? You end up with offspring with the parent and both parent phenotypes and a recombination of the two, yellow, wrinkled, green, round. Okay? Alright, so like we looked at with the flies, crossing over has something to do with how that recombination occurs. And they were able to calculate the frequency of recombinants that they received. Okay? In these diagrams, recreating the test cross from the previous figure with Morgan's Drosophila, we track chromosomes as well as genes. The maternal chromosomes were color-coded red and pink to distinguish one homolog from the other before any meiotic crossing over has taken place. Because crossing over between the B and VG loci occurs in some, but not all, egg-producing cells, more eggs with parental type chromosomes than with recombinant ones are produced in the mating females. Fertilization of the eggs by sperm of genotype homozygous for black vestigial wings, okay, gives rise to some recombinant offspring. The recombination frequency is the percentage of the recombinant flies in the total pool of offspring. Okay? Here's showing your recombinant frequency. It's 17 here. So that only takes a look at, alright, meiosis 1 and 2, if you have a homozygous male, no recombination will take place after replication because there's nothing to exchange. It's exchanging the same genes. Okay? However, your heterozygous chromosomes pairs, after replication, they can exchange a segment, this is crossing over, a segment of genes with their homologous pair. So when your genes are spread out into your offspring, into your gametes, so will turn into the offspring later, you end up with eggs with each of a parent type and a recombinant type. So when you look at the numbers arising in the F2 population here, this would be your F2s, parental type to offspring type, you had 331 total recombinants over 2,300 total offspring. So when you divide that out, it's only 17%. Okay? Alright, mapping the distance between genes using recombinant data. This is discussed on, make sure I tell you the right page, on 296, if you want to look at the figure. Okay? Alfred Sturveant, one of Morgan's students, constructed a genetic map, an ordered list of genetic loci along a particular chromosome. He predicted that the further apart two genes are, the higher the probability that a crossover will occur between them, and therefore the higher recombination frequency. Alright, a linkage map is a genetic map of a chromosome based on recombination frequencies. Distance between genes can be expressed as map units. One map unit, or centimorgan, represent 1% recombination frequency. Map units indicate relative distance in order, not precise location of genes. Okay, so this is showing you that figure on 296. Alright, results found. The observed recombination frequencies between three Drosophila gene pairs, BCN, which is B to CN, which is 9%, and CN to VG, which is 9.5%. Okay? and then B to VG, which is 17 total percent, are your recombination frequencies, okay? They best fit a linear order in which CN is positioned about halfway between the other two genes. Alright, the BVG combination frequency, 17%, is slightly less than the sum of B to CN and CN to VG frequencies. That would be 18.5%. Because the few times that crossovers occur between B and CN, an additional crossover occurs between CN and VG, the second crossover would cancel out the first, reducing the observed B to VG recombination frequency while contributing to the frequency between each of the closer pairs of genes. Alright, the value of 18.5% or 18.5 map units is closer to the actual distance between the genes. So a geneticist would add the smaller distances in constructing a map. Okay? Genes that are far apart on the same chromosome can have a recombination frequency near 50. Such genes are physically linked but genetically unlinked, and behave as if found on different chromosomes. Sturgeon used recombination frequencies to make linkage maps of fruit fly genes. Using methods like chromosomal banding, geneticists can develop cytogenic maps of chromosomes. Cytogenic maps include the positions of genes with respect to chromosomal features. Okay, going into 15.4, we'll keep rolling, try to knock this out. Abnormal chromosome number can cause non-disjunction. So during meiosis 1, non-disjunction can occur. Your pairs of homologous chromosomes do not separate normally during anaphase 1. So as a result, one gamete receives 2 of the same chromosome and another gamete receives no copy. Okay, so this is showing that non-disjunction here. Your chromosomes here separate. Here's where you're. Alright, your chromosomes here do not separate appropriately, so instead of having one of these homologous pairs, instead of having one group of sister chromatids in each of the daughter cells at the end of meiosis 1, you have 3 pair going in to meiosis 2 versus 1 pair. So you end up with 3 chromosomes, 3 sets of chromosomes versus 1 set. Okay? So this is non-disjunction of homologous chromosome in meiosis 1. This is when it occurs in meiosis 2. Everything separates appropriately through meiosis 1. However, when you get to meiosis 2, this pair of sister chromatids does not split, so you end up with a cell that has 3 pair versus the partner that has 1. Okay? So this shows the non-disjunction in meiosis 2. So this creates an issue because this gamete has an incorrect chromosomal number as well as this gamete. Okay? And these gametes all have incorrect chromosome number. So that can start to introduce genetic defects. Okay? Aneuploidy results from the fertilization of gametes in which non-disjunction has occurred. Offspring with this condition have an abnormal number of particular chromosomes. A monosomic zygote has only 1 copy of a particular chromosome. Okay, mono means 1. A trisomic, tri meaning 3, has 3 copies of a particular chromosome. Okay. Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. Triploidy, 3N, is 3 sets of chromosomes. Tetraploidy, 4N, is 4 sets of chromosomes. Polyploidy is common in plants, but not animals. Polyploids are more normal in appearance than aneuploids. Okay? So remember, aneuploids is fertilization of gametes where non-disjunction has occurred. Polyploidy is where the organism has more than 2 complete sets of chromosomes. Okay? Alright, alterations of chromosome structure cause versus breakage. Alright, breakage of a chromosome can lead to 4 types of changes in chromosome structure. You can end up with deletion, duplication, inversion, or translocation. Deletion, just like it sounds, removes a segment of the chromosome. Duplication repeats a segment. Inversion replaces a segment within a chromosome, and translocation moves that segment from one chromosome to another. Okay? Here is showing you some examples. Alright, this is when breakage occurs. If you have deletion of part D, okay, deletion of part D, here's your resulting chromosome. Duplication of the BC region here. Inversion, it goes BCD, here would be DCB. It flip-flops the order of those loci. Alright, reciprocal translocation, A, B, and M and O are flip-flopped here. Okay? Thus altering both chromosomes. Alright, Down syndrome is trisomy, meaning 3 copies of chromosome number 21. Down syndrome is an aneuploid condition that results from 3 copies of chromosome 21. It affects about 1 out of every 700 born in the United States. The frequency of Down syndrome increases with the age of the mother, a correlation that has not yet been explained. Alright, here's showing what a chromosome or a karyotype with someone with trisomy 21, what they look like. Okay. Aneuploidy of sex chromosomes. Non-disjunction of sex chromosomes produces a variety of aneuploid conditions. Klinefelter syndrome is the result of an extra chromosome in a male, producing XXY individuals. Monosomy, which is X, called Turner syndrome, produces X0 females who are sterile. It is the only known viable monosomy in humans. Most other conditions that cause monosomy in the autosomal cells are going to be lethal, and most of the time a fetus that has a monosomy in a certain cell won't survive to birth, let alone be born. Okay? Disorders caused by structurally altered chromosomes. The syndrome, alright, forgive my French, but it is cri du chat, or cry of the cat, results from a specific deletion in chromosome 5. A child born with this syndrome is mentally retarded and has a cat-like cry. Individuals usually die in infancy or early childhood. Alright, certain cancers like chronic myelogenous leukemia or CML are caused by translocation of chromosomes. Alright, that's just a few of the many different disorders that can be caused by non-disjunction occurring in chromosomes. But like it stated, there are many other issues that occur with this non-disjunction, with having too many or too few sets of chromosomes at a certain point in their cells. A lot of those don't survive to birth. Those cells are set to go through apoptosis and not continue replicating, so that will cause that, if it is a devastating genetic disorder, it will cause spontaneous abortion, which would be miscarriage. A lot of times miscarriage occurs either due to an issue with implantation or issues with genetic disorders. That can cause miscarriage. Okay, the baby can't grow functionally, then it won't be able to survive long enough to grow to term. All right, and last section, 15.5. Some inheritance patterns are exceptions to the standard chromosome theory. There are 2 normal exceptions to Mendelian genetics. One exception involves genes located in the nucleus, genomic imprinting. The other exception involves extranuclear DNA, genes located outside the nucleus in the mitochondria and chloroplast. Okay? Genomic imprinting, variation in phenotype. For few mammalian traits, the phenotype depends on which parent passed along the alleles for those traits. Such variation in phenotype is called genomic imprinting. It involves the silencing of certain genes that are stamped with an imprint during gamete production. Alright, so this figure here, on which you can locate yourself on page, I believe it's 301. Let me double check for you. Yes, 301. Alright. This is showing mating between wild-type mice and those homozygous for the recessive mutant IGF2 allele produce heterozygous offspring. Okay, here are your heterozygous offspring. Okay, you start with your homozygote. You have your heterozygous offspring. All right, the mutant phenotype is seen only when the father contributed the mutant allele because the maternal allele is not expressed. Okay? So, what this is looking at, the normal IGF2 alleles expressed on the father's chromosome, okay? The normal IGF2 allele is not expressed on the maternal or the mother's chromosome, alright? Your wild-type mouse that goes into the cross is normal sized, okay? This mouse here has the mutant IGF2 allele inherited from mom, but it is not expressed because it receives the normal IGF2 allele from dad that is expressed. This mouse, however, receives the mutant allele from dad, normal from mom, and it is expressed, and so the mouse is dwarf. Okay? This phenomenon is different from sex linkage. Most imprinted genes are on autosomes. Okay? Let's see. I wanted to add one more thing. Genomic imprinting occurs during formation of gametes and results in silencing of one allele of certain genes because the genes are imprinted differently in the sperm and the egg. A zygote expresses only one allele of an imprinted gene, either the allele inherited from mom or from dad. Okay? The imprints are transmitted to all body cells during development, so either the maternal or paternal allele of a given imprinted gene is expressed in every cell of that organism. In each generation, the old imprints are erased in gamete-producing cells, and the chromosomes of the developing gametes are newly imprinted according to the sex of the individual from the gametes. In a given species, the imprinted genes are always imprinted in the same way. For instance, a gene imprinted for maternal allele expression is always imprinted for maternal allele expression, generation after generation. Alright, so let's look at our little mice here. Consider, for example, the mouse gene for insulin-like growth factor, IGF2, one of the first imprinted genes to be identified. Although this growth factor is required for normal prenatal growth, only the parental allele is expressed. Evidence that this gene is imprinted initially came from crosses between wild-type mice and dwarf mice homozygous for a recessive mutation in the IGF2 gene. The phenotype of heterozygous offspring, which had one normal allele and one mutant, differed depending on whether the mutant allele came from mother or father. So, this gene is whatever the paternal gene is what will be expressed in this case. Okay? Alright, it appears that mammalian genomic imprinting or gene silencing is the result of methylation of DNA, the addition of a CH3. Genomic imprinting is thought to affect only a small fraction of mammalian genes. Most imprinted genes are critical for embryonic development. Alright, the inheritance of organelle genes, your extranuclear DNA. Extranuclear genes, or cytoplasmic genes, are genes found in organelles in the cytoplasm. Your mitochondria and your chloroplasts and other plant plastids carry small circular DNA molecules. Extranuclear genes are inherited maternally because the zygote cytoplasm comes from the egg. Okay? The first evidence of extranuclear genes came from studies on the inheritance of yellow or white patches on leaves of an otherwise green plant. Okay? This DNA that is inherited through your mitochondria and your chloroplast is explained like maternal DNA, because it is only inherited through the cytoplasm of the egg cell, because your sperm doesn't donate any cytoplasm, negligible amounts. So this small circular DNA that's inherited through mitochondria and chloroplast are strictly genes that you receive from your mother. Okay? So that is it, guys. We have finished with chapter 15. Make sure you review over the things that you should now know, and make sure you're able to answer those questions. Those are good questions to prepare for our test when we get to the end of chapter 17. So 16 to come this week, 17 after spring break. Alright, please let me know if you have any questions. Make sure that you've answered the checkpoint questions for this chapter. Thanks, guys. Bye.